Hello and a very good evening to all our viewers who are joining me in this live interaction on Facebook and YouTube page of News9. And today I will be speaking about another discovery made by scientists which will probably help us get through this pandemic in the coming days or in the near future. Well, now if I have to give you a brief introduction, clinical trials are testing convalescent plasma which contains antibodies from people who already had COVID-19 as a treatment for those who are currently battling the infection. But there isn't enough plasma for those who might need it, and it isn't proven to work yet. So in the latest, the scientists of University of Pittsburgh School of Medicine have isolated the smallest biological molecule that completely and specifically neutralizes the SARS CoV2 virus, which is the cause of COVID-19. And this particular antibody which they extracted and isolated is a component that is close to 10 times smaller than a full-sized antibody and has been used to construct a drug known as AB8 for potential use as a therapeutic and also prophylactic against SARS CoV2. So the drug AB8 is highly effective in preventing and treating coronavirus infection in mice and also hamsters. Now these claims were made based on results from a test which was conducted on mice and hamsters using this particular drug which they extracted from the smallest component of an antibody. So the tiny antibody component is the variable or heavy chain domain of an immunoglobulin, which is a type of antibody found in the blood. So it is found by fishing in a pool of more than 100 billion potential candidates using the SARS CoV2 spike protein as bait. And AB8 is created with the VH domain and is fused to part of the immunoglobulin tail region, adding the immune functions of a full-size antibody without the bulk. Now, in order to answer a few questions and also give us a little more insights about this, we have with us Dr. Sitesh Roy, and many thanks, Doctor, for joining me in this live interaction. And my first question to you is, do you think that this particular discovery will change the course of vaccine development? Earlier, it was just about generating antibodies, but now it has been reduced to a specific component of the antibody, which is very crucial in defeating the COVID-19, Doctor.
So thank you, Deepak. I think that the vaccine development will need to continue in the way it is, because the way a vaccine works is it generates antibodies and cytotoxic T cells and provides a much longer immunity that we are really seeking against the SARS CoV2 virus to protect people from the COVID-19 infection. This finding, however, with the fraction of an antibody, so the heavy chain is, you can say, 1/4 of an antibody molecule, and even just the variable part of it is how we get to hardly 1/10 the size. If this antibody, which they have found, very efficiently binds to the three what are called S proteomers of the SARS CoV2, which the virus requires that to bind to the ACE2 inhibitor and get into the cell. So if we inject or nasally administer this tiny antibody to humans who are either infected or at risk of getting infection because they were exposed to people with it very closely, that's how the prophylactic and the therapeutic application comes in. Then those people in their body could bind the virus very efficiently by this AB8 construct molecule and prevent the infection, and also help progression of the infection by preventing more and more cells from getting infected by the virus. So I truly feel that this is an amazing application of the field of immunology and all the advances that we have in it, where fractions of molecules can be made using something called a bacteriophage, which is something that can be used to replicate these molecules and then do the fusion technology to create a construct that can block and interfere with the SARS CoV2 virus and prevent the infection. So I think vaccines are in their place and antibody therapy is in its own place, and both are very, very important steps in our battle against COVID-19.
So, doctor, do you think that this particular discovery could be a game changer in the current situation?
I certainly believe that, right now this has been very successful in the mice and hamster studies, where doses as little as 2 milligram per kilogram body weight have been extremely successful in preventing or fighting off the infection. So I believe that once we get into the human safety and dosing trials and the human phase 1, phase 2, phase 3 trials, and we get good results, it could really make a very big difference should it be just as successful in humans as it has been in animals. So I believe it has the potential to be a game changer, because certainly the science behind it is very, very sound. And also this molecule, when tested against 5,300 human proteins, did not seem to interfere or interact with them, which makes it very safe and unlikely to cause side effects in the human body. So I certainly will be very eagerly looking at the next phases of development of this molecule.
Also, doctor, now how is this tiny component of an antibody, which is of a full size antibody, more effective than the bigger half of it?
So what ends up happening is the variable region of an immunoglobulin molecule is where the, you can say, flexibility and malleability of an antibody occurs to bind to different parts of the viral proteins. And this section we could never really separate and use in the past because the antibody comes as a whole. It's an entire structure. There are two heavy chains, there are two light chains, and then there is a fixed segment, the FC segment that they have described as the part of VH, VHFC and AB8 antibody. And so being able to separate that out, to construct it separately and attach it to just the necessary portion, really reduces the size. By reducing the size of the antibody, the advantage is that not just you may require less to be given to the patient. So for example, in a plasma infusion, in a donor plasma infusion, where you're giving a patient who is currently battling COVID-19 the plasma of someone who has recovered, you have to give 250 ml of their plasma. And even in that, if the antibody, the neutralizing antibody titer is not adequate, then you may not get a good response, which is why the trials are failing, because the neutralizing antibody titer is not being as adequately checked before the infusions are given. And so all of those issues are eliminated when you have a tiny antibody, which is a fraction the size. You need less quantity. You can give it also potentially, they are saying, through a nasal route without an infusion or injection. But that is still to be studied and seen whether that will be an adequate dose. So there are multiple advantages on how such a small antibody, by only binding. So we've all heard of the spike protein and how the spike protein is so necessary to bind to the ACE2 inhibitor. These tiny antibodies go directly and bind the three exact points. So like a jigsaw puzzle that attaches in that jigsaw puzzle, if it goes and catches the 3 points on the jigsaw that are required for the puzzle to fit, the virus being one and the human cell being the other, the ACE2 inhibitor, if it fits right there where the jigsaw won't fit, you have blocked the infection and the entry of the virus into the cell.
Okay. Also, doctor, now the response, do the response differ between the tiny antibody particle and the full size antibody? How do they react when exposed to a virus? Do they react in a similar way, or is there a difference between their reactions to a virus, doctor?
So I think in this, the key again is the way the molecule is constructed, because the variable chain is where all the structural adjustments are done by the human immune system, so that it is a good fit in that jigsaw puzzle to what the virus carries on its spikes. I think that the tiny construct AB8 should fit and work just as well as the large antibody, the entire antibody does. So I don't think that there should be a problem. But again, like I said, testing in mice and hamsters is one thing. and testing it in humans is completely another thing. So I think we will require more studies. We have some in vitro data, but we have to see in vivo. In vitro is where you do it in a lab. In vivo is where you actually give it to a human being and see how they respond. So I think we are all eagerly awaiting that. Those are not yet done.
Also, doctor, now if we see the light of the day using this particular tiny component of a full-size antibody, how will it be administered to humans while during the vaccination phase?
So again, Deepak, I think the vaccination will be a separate process. This antibody treatment will be in the therapeutic realm. So like in therapy, we have a drug like remdesivir that is used in moderate to severe infections. We have a drug like tocilizumab that is used in a cytokine storm. Similarly, when a person were to get the COVID-19 infection and they were to be admitted to the hospital, that is when they could be given this antibody through a nasal spray or through an injection or an IV infusion. That still will depend on the dosage that is being required in the human body. So that is why the dosing studies, which is the phase 1 trials, will tell us what will be the route of administration. And the vaccination protocol will continue in its own way once we have an immunogenic, successful, safe vaccine. And the therapeutic, the treatment part with this tiny antibody will progress in its own way for those who are infected. And for example, if a person is infected and his family members are very closely exposed to him, or someone at work or at a hospital were to get infected and their colleagues were very closely exposed, then even prophylactically, a person preventively could be given this tiny antibody fragments and block the entry of the virus into many cells in their body and prevent the infection completely. The prevention has been shown in hamster studies. We now will have to wait to see what it does in human studies, though those studies will be quite tricky to do, I can tell you.
Now, doctor, since clubs, metros and schools are also set to reopen, until we see a vaccine, a concrete vaccine being developed, what is your message and how do you urge the people to follow all the safety protocols, doctor?
Deepak, we have spoken about this before also, and I will say it again. Even if we have a safe, successful immunogenic vaccine, it will be at least 2 years before each and every person who needs to get it will get it, maybe even longer, we don't know. So in the meantime, till the virus itself slows down or mitigates, or enough people have had the infection, which is highly unlikely that we develop herd immunity before a vaccine, the precautions are 100% essential. People need to wear their face mask and wear it correctly when they are around even 1 or 2 other people. They need to continue to hand wash and sanitize their hands if they are touching surfaces other than within their own home, unless someone in their home is sick, and then they may need to do it at home also. And lastly, maintaining physical distancing, at least 3 feet if you're talking to people, and 6 feet under ideal circumstances in most scenarios, are still extremely important for breaking the chain of spread of this virus through respiratory droplets, whether they are macro or micro droplets. So all the precautions absolutely need to be continued to be followed, and people should not let down their guards. We are not there yet.
Right, doctor. Thank you for that message. Well, there you heard it. That was Dr. Sitesh Roy giving us his insights about the tiny component of an antibody which was extracted and isolated by a group of scientists, and it proved to be effective in mice and hamsters as well. But the test on mice and hamsters is completely different, and when it comes to humans, it is a huge challenge. So like the doctor said, we might get a vaccine in the next year, or it may even take a couple of years. And even the Serum Institute of India has said that we might see a vaccine only by 2024. So until then, like the doctor mentioned, all the safety and precautionary measures are important because that is the only weapon that we have against COVID-19. So make sure you equip all of your weapons and also fight and defeat this COVID-19 by following all the safety and precautionary measures. Until next time, take care and stay safe.